Hi there, scientists-to-be. I'm the Geneticist, and welcome back to the lab. Today, we're diving into the Polymerase Chain Reaction, or PCR, a true rock star of molecular biology. Are you ready? So what is PCR? Simply put, PCR is a way to make millions, even billions, of copies of a very specific piece of DNA from even a tiny, tiny starting sample. This process involves a series of precise temperature-controlled steps that enable the exponential replication of a target DNA region, making it detectable and analyzable. This fundamental biotechnology technique lets us make millions of copies of a specific piece of DNA, even from a tiny sample. Think of it as a super awesome DNA copy machine that's essential for everything from crime shows to disease diagnosis. So what do you need for this DNA party? Just a few key players you mix together in a reaction tube. Number 1, DNA template. Your original DNA, the tiny bit you want to copy. If it's RNA, like from a virus, we first convert it to DNA with real-time PCR. Number 2, primers. These are short, custom-made DNA navigators that tell the copying enzyme exactly where to start and stop. Designing them perfectly for specificity and melting temperature is crucial for accuracy. Number 3, DNA polymerase. Our master builder enzyme, usually Taq polymerase, which assembles new DNA strands. Number 4, nucleotides A, T, C, G, the individual DNA building blocks. Number 5, buffer to keep everything stable and happy for the reaction. All these ingredients go into a thermocycler, a machine that rapidly changes temperatures through cycles, usually 25 to 35 of them. Each cycle has 3 main steps. First, denaturation, 94 to 96 degrees Celsius, 20 to 60 seconds. We crank up the heat to unzip the double-stranded DNA template into 2 single strands. Second, annealing, 50 to 65 degrees Celsius, 20 to 60 seconds. We cool it down just enough for our primers to bind to their specific spots on each single DNA strand. This is where they anneal. Third, extension, 72 degrees Celsius, 30 to 60 seconds per kilobase. The perfect temperature for Taq polymerase to get to work. It starts from the primers and builds new complementary DNA strands using the free-floating nucleotides. After these cycles, we do a final longer extension step, like 72 degrees Celsius for 5 to 10 minutes. To make sure all DNA fragments are fully complete, then cool it down to 4 degrees Celsius for storage. What's incredible is that each new strand becomes a template for the next cycle. This means the amount of DNA theoretically doubles with each round. After one cycle, one DNA molecule becomes 2. After 30 cycles, you get over 1 billion copies of your target DNA. This exponential power makes PCR incredibly sensitive, allowing us to detect DNA from even the tiniest samples, from a single hair to ancient mummy DNA. Of course, the amplification eventually plateaus as reagents run out, but by then you've got plenty of copies. Once the reaction is done, we usually analyze the PCR products using gel electrophoresis. This lets us confirm the presence, size, and specificity of the amplified DNA, making sure our copying machine worked exactly as intended. PCR rules the lab and is a superstar in so many fields. 1. Disease diagnosis, quickly identifying viruses like COVID-19 and bacteria. 2. Genetic testing, diagnosing inherited diseases. 3. Forensics, amplifying DNA from crime scenes. 4. Research, cloning genes, studying gene expression. 5. Paleogenetics. Like in our Decoding Dynasty series, it's essential for studying ancient DNA. And for even more power, there's real-time PCR, or Qpcr. This clever technique lets us quantify the amount of DNA as it's made using fluorescent dyes. It's super accurate for measuring how much virus is in a sample or how active a gene is. We even have single-cell RT-qPCR to look at gene expression in just one cell. Now, for you future lab gurus, getting PCR just right is an art and a science. Optimizing PCR amplification is crucial for super-sensitive, specific, and reproducible results. It's not just throwing ingredients in a tube. Number 1, primer perfection. You want primers that bind to conserved regions of your target DNA to avoid genetic mutations throwing off your results. Scientists constantly monitor genetic changes in organisms to ensure their PCR tests stay accurate over time. Number 2, cycle count. Too few cycles, and you won't get enough DNA. too many, and you risk getting false positives or amplifying unwanted DNA. It's a delicate balance. Number 3, template quality. Using high-quality DNA from the start makes a huge difference in sensitivity and yield. Sometimes even direct PCR from things like whole blood can work, streamlining the process. Number 4, validation is key. Especially in diagnostics, we often validate PCR results with other methods to confirm sensitivity and make sure it's detecting exactly what it should. And sometimes we use multiplex PCR to detect multiple targets at once. Super efficient, but needs careful setup to avoid crosstalk. So while PCR is powerful, knowing how to fine-tune it is what separates a good reaction from a great one. So the next time you hear about DNA evidence, a new medical test, or even ancient mummy DNA, give a little nod to PCR and its amazing multi-step dance. It's a testament to how clever biotechnology can unlock revolutionary insights in science and medicine. You've just decoded one of genetics' most fundamental techniques. Now you know the secret behind making billions of DNA copies and even counting them. Pretty cool, huh? If you enjoyed this video, hit that like button, share this video with your fellow scientists-to-be, and definitely subscribe to The Geneticist for more awesome adventures into the world of genes and beyond.